Hello dear students, welcome back once again to Kamala Online Learning. We are back again with another informative video lesson from your schedule. Please be completely attentive during the teaching. Keep your textbook and also a rough book with you. Note down important points and difficult or new words as we go. This video lesson will begin with an overview which will give you an idea about the contents of the lesson. It will be followed by learning objectives. Please note down the learning objectives in your notes and try to answer them at the end of the session. If you have any doubts, rewatch the video and refer to your textbook and study material. In case you need further clarification, please mail your doubts to kamalaonlinelearning@gmail.com. This video is recorded with simple, easy to understand language and eye-catching graphics. We hope it helps you to understand the concepts better. So, let's get started.
Theory of Microorganisms, Part 2.
Learning objectives. By the end of this chapter, you will be able to
know useful and harmful microorganisms. Know the discovery of disease-causing microorganisms. Explain the discovery of vaccine for diseases. Interpret the microorganisms and soil fertility. Know the harmful microorganisms. Understand the common diseases caused by microorganisms. Explain the disease-causing microorganisms in plants and animals. Explain food poisoning and preservation of food. Describe the process of pasteurization. Understand the importance of storage and packing of food. Explain the nitrogen fixation in plants.
Introduction. Before entering into the chapter, follow the instructions shown on the screen.
From the previous chapters, we know that the Earth's biosphere comprises of a large number of organisms. These organisms can be further classified into various groups and categories. The microorganisms are invisible to the naked eye and are part of the organisms. Microorganisms are present all around us, that is soil, water, air, within the bodies of animals and plants, etc. Microorganisms influence their surroundings in many ways. Do you know what are the uses of microorganisms? Are microorganisms used for medicinal purposes? Explain the commercial uses of microorganisms. List the various types diseases caused by harmful microorganisms. What is food poisoning? Define pasteurization. How can you store and pack the microorganisms? Let us try to answer these questions and learn more about the study of microorganisms, their uses, harmful effects, and inventions using them. Microorganisms are present in all the possible places on the earth, that is in the air, water, food, soil, etc. They are even present in our bodies, in plant bodies and others. Some of these are extremely useful, while some of them are not, and some are dangerous as well. Thus, microorganisms prove to be equally useful and harmful. They can even cause the death of people and living beings sometimes. Let us learn about useful microorganisms. We use some of the microorganisms in our daily life. For example, making of curd, preparation of wine, preparation of cake, bread, idli, dosa, etc. Some microorganisms are used in the preparation of medicines which are required to cure different types of diseases. Some other microorganisms are used to make fertilizers which makes the soil fertile and yield more crops. Let us conduct a few activities to know more about these. Here, let us perform an activity for making of curd. Take some lukewarm milk in a jar. Take 2 bowls. Pour the milk in both the bowls. Now, add a little curd in one of the bowls. Keep the 2 bowls in a warm place for 5 to 6 hours without disturbing. After 5 to 6 hours, when we observe both the bowls, the bowl in which curd is added converts to curd, while the bowl in which nothing was added remains as milk. From the above activity, we have noticed that the milk which is turned to curd comprises of a bacterium called lactobacillus. This bacteria serves as an agent and causes milk to get converted into curd. Curd is a part of everybody's meal, so this is one of the domestic ways where microorganisms prove to be helpful. Take 100 grams of maida powder in a bowl. Add 2 or 3 spoons of yeast powder to the maida. Now add water to the bowl. Knead the powder to make it into a dough. Keep the dough in a warm place for 3 to 4 hours. After 3 hours, it is observed that the kneaded dough has expanded and increased in size. It has risen above its previous level. From the above activity, we have noticed that the dough is increased when yeast powder is added to it, because the yeast powder causes the process of fermentation. During fermentation, carbon dioxide gas is produced, causing the bubbles of gas to fill the dough. Hence, the volume of dough increases and makes it spongy in nature. This process is applied in the preparation of fermented foods like idli and dosa. Take 2 beakers and pour water into them. Now, add 5 to 10 spoons of sugar in both the beakers. Next, add 2 to 3 spoons of yeast powder to one of the beakers. Close the lids of the beakers and keep them in a warm place. Leave the beakers for 3 to 4 hours. When we smell the contents of the beakers, the beaker in which yeast powder was added would emit a fruity smell. The beaker with simple sugar solution would smell normal. The beaker into which yeast powder was added will emit a fruity smell because the sugar solution is converted into alcohol. The process of conversion of sugars into alcohol is called fermentation. The fermentation process is widely used in the production of alcohols, beer, and acetic acid on a large scale. Yeast is produced from natural sugars. The natural sugars are present in the grains of pulses like wheat, barley, rice, and also from crushed fruit juices like grapes. The ethyl alcohol is prepared by adding yeast to the fermentation process, in which molasses are used, which is produced in the sugar industry. Let us learn about medicinal uses of microorganisms. Sometimes we may fall ill, get injured, or even need to undergo an operation. During these circumstances, doctors prescribe certain medicines to kill the bacteria involved. These medicines that kill the bacteria are called antibiotics. They are produced by growing specific microorganisms. Nowadays, antibiotics such as streptomycin, penicillin, erythromycin, and tetracycline are majorly produced. Antibiotics are used for curing bacterial diseases like typhoid, diphtheria, leprosy, cholera, etc. They are also used for the control of diseases caused by the bacteria in plants and animals. Antibiotics prescribed by a qualified doctor ought to be used, otherwise it could harm us. Using the antibiotics unnecessarily affects the blood cells which fight against infections and leads to lack of immunity. There are chances of killing useful bacteria that are present in the intestines, causing the increase in the resistance towards the antibiotics. Excess usage of antibiotics leads to the weakening of body. Dr. Alexander Fleming discovered the antibiotic penicillin. He was an army doctor and observed the deaths of many injured soldiers due to bacterial infection on the wounds. While working on antibiotics in his lab, he was growing bacterial colonies in the petri dish and observed that a particular fungi or mold was preventing the growth of the bacteria. He separated them and tested this mold on other disease-causing bacteria and observed that the mold was capable of killing other diseases-causing bacteria as well. The mold or fungus which killed the bacteria was recognized as Penicillium notatum. The substance that was produced by the fungus Penicillium was named as penicillin. Dr. Alexander Fleming named the substances that killed the bacteria as antibiotics. The discovery of penicillin antibiotic was announced in 1929, and in the year 1945, Dr. Alexander Fleming was awarded the Nobel Prize. The discovery of penicillin gave way for many other discoveries of antibiotics like streptomycin, erythromycin, etc. All of these have made a very significant contribution to the medical field. Let us study about the inventor of aromycin. Dr. Yellapregada Subbarao, who belongs to West Godavari district of Andhra Pradesh, India, is the person who made the discovery of the antibiotic aromycin or tetracycline. Today, this antibiotic is used for curing many bacterial diseases like typhoid, cholera, plague, tuberculosis, etc.
Most commonly, we get various kinds of syrups, tablets, etc. prescribed by the doctors in case we are prone to illness or some diseases. Then, why do you think vaccination is done in case of other disease like polio? This is because vaccination protects us and makes us resistant towards a particular disease for a longer duration of time, possibly for the entire lifetime. You must have heard about the importance of vaccinating children who are less than 5 years age to prevent polio. Were you also vaccinated against polio in your childhood? In 1956, our government took up a program called Pulse Polio to eradicate polio from the Indian society. In 1952, Dr. Jonas Salk discovered the vaccine for polio. He desired to distribute his discovery freely to everyone, hence never patented his polio vaccine. Oral polio vaccine was discovered by Dr. Albert Sabin in the year 1957. Our body produces some defenders to fight against the disease-causing microorganisms whenever they enter our body. These defenders are called antibodies and fight the disease-causing microorganisms. The vaccine contains weak microorganisms, so whenever it is injected into our body learns to defend itself by producing the antibodies. Hence, when the disease-causing microorganisms enter the body, the existing antibodies fight and eliminate them. Therefore, the disease gets controlled. The weakened disease-causing microorganisms injected into our bodies are called vaccines. Polio vaccine is given orally, while most other vaccines for diseases such as smallpox, chickenpox, hepatitis, tuberculosis, etc. are given in the form of injections. At present, we have numerous vaccines to protect ourselves from several disease-causing microorganisms. But 300 years earlier, there were no vaccines. Many scientists experimented in the 18th century for the discovery of vaccines. One of the greatest contributions was made by Dr. Edward Jenner of United Kingdom in the year 1796. Rabies is a deadly disease that causes brain inflammation and ultimately death. The rabies virus enters the body when a dog which is having the same infection bites us. Louis Pasteur in the year 1885 discovered a vaccine for rabies. Nowadays, vaccines are made on a large scale from microorganisms to protect human and other animals from several diseases.
Dr. Edward Jenner is called the father of immunology. He decided to set up his medical practice in a village, which helped to discover the vaccines and help the mankind from extinction with diseases. In his observation, he found that milkmaids developed a less serious disease called cowpox, but not a deadly smallpox. He understood that this was because the milkmaids started to develop a kind of resistance against cowpox. In the year 1796, Jenner took the fluid out from a milkmaid's cowpox pustule and inoculated an 8-year-old boy with the permission of his parents. After 6 weeks, the boy was exposed to smallpox, but no symptoms of smallpox were developed in the boy. This great invention of Jenner saved the lives of millions of people who were affected with this disease. This invention led to the discovery of many other vaccines to protect people from various other harmful diseases. In Latin word, cow is called vacca and cowpox is called vaccinia.